SwayamPrabha. Digital India. Educated India.
Dear students, you are welcome in this class. I am Dr. Dinesh Kumar, Principal Scientist from Division of Agronomy, ICAR, Indian Agricultural Research Institute, Pusa Campus, New Delhi. This lecture pertains to the course Agronomy of Oilseed, Fiber and Sugar Crops. The title of the lecture is Insect-pests, Diseases and Post-Harvest Management of Cotton and Recommended Cropping Systems for this crop. The learning objectives are to describe the management of insect pests and diseases of cotton crop. This crop particularly is affected by number of insect pests and diseases. So we will discuss their symptoms, how they affect or damage this crop, and then what are the management options to reduce the incidence of insect pests and diseases in cotton. And then we will explain to you the harvesting methods and post-harvest management of the cotton produce, and it will also include some quality traits. Then to detail the recommended cropping sequences included in cotton under different parts of the country. So let us check some glossary, some difficult words that could be useful to you. Foliar feeding, so it has got several meanings. Out of those, two meanings are: number one, leaf consumption, usually by caterpillars or other insects. Second is to apply foliar spray to the plants. Then another term is frass. It is a term applied to insect feces, or in fact it is excreta, the shape of which is sometimes used in family or species level identification, also called fecal pellets or dropping. So this is called as frass. Then comes the fungicide, a chemical which controls or reduces the growth of fungus is your fungicide. Then light trap. These are used for the management of insect pests, and these are actually devices consisting of at least an ultraviolet light which is attractive to night flying insects, and a collection of container used to monitor the timing and relative abundance of selected insect species. Then pheromone trap that uses either a natural or synthetic insect sex attractant, pheromone. So these traps are usually species specific. Then let us see what are major insect pests of cotton and what kind of damages are being done in this crop. So having succulent leaves, attractive flowers with nectar and fruits of different sizes, cotton attracts number of insects. It catches a complex of insect pests right from sowing, vegetative growth phase, flowering and reproductive phase. You will learn in this lesson. So at every stage of this crop, you will find some or other insect pests that damage this crop. So continuously this crop suffers from invasion by insect pests. Among these pests are the bollworm complex, which include 3-4 kind of bollworms which attack the bolls, the reproductive unit of cotton, and they pose great threat to cotton, followed by the sucking pests that suck the sap from different parts of the plant. The sucking pest complex includes a variety of aphids, jacids, thrips and bugs. So a student can see that these are the sucking pests: aphids, jacids, thrips and bugs. And over 160 species almost of insects are known to cause economic losses or damage to the cotton crop in different production systems in India, even in many other countries also. Now let us classify these insect pests into 3 groups. One is bollworm complex. So it includes number of bollworms like American bollworm, quite destructive insect. It is known as Helicoverpa armigera. Other is spotted bollworm, Aereus insulana, Aereus vitella, and pink bollworm, Pectinophora gossypiella. So students are advised to remember their names and also the scientific name of these bollworms. Then comes the sucking pest like leafhopper, Amresca bigutula, and then aphid, Aphis gossypi, thrips, Thrips tabacea, whitefly, Bemisia tabacea, cotton mealybug, Phenacoccus solenopsis, and then also papaya mealybug. Means mealybug of papaya can infest the cotton crop. So this is your Paracoccus marginatus. Paracoccus marginatus is your papaya mealybug. Now third category is grouped as other insect pests. So it may include your Indian cotton mirid bug, this mirid bug, and then stem weevil and tobacco caterpillar, Spodoptera litura, are also categorized as major or key pests, especially in South India. So by now you could understand that what are the major insect pests that affect this crop, mainly your bollworms, mainly your sucking pests, and also certain weevils, certain caterpillars affect this cotton crop. Indian cotton ecosystem experienced a phenomenal change in its pest profile since introduction of BT cotton in 2002, that is Bollgard 1 and Bollgard 2 was introduced in 2006. So what was happening, these bollworms were doing a lot of damage to the cotton crop, but this BT cotton was introduced in India 2002, and then these insects were easily controlled, these bollworms were easily controlled, and later on means the productivity of cotton in our country increased tremendously, and farmers were using less insecticides to control the insect pest in cotton crop. But with the passage of time, year to year, then some minor pests emerged and they become the major pests. Sometimes it happens. BT cotton provided protection against bollworm and more effective against Heliothrix armigera. Also offered protection against spotted and pink bollworm. So these were the major pests, and cotton crop almost, particularly BT cotton, escaped the damage by these kind of bollworms. From 2014 onwards, high incidences of pink bollworm were recorded on Bollgard 2, substantiating resistance development to both genes employed in Bollgard 2. So what happens with the passage of time? The resistance of hybrids of BT cotton is diminished, it is reduced with time. Widespread infestation of pink bollworm on BT cotton has been reported from cotton Indian states, and there have been incidences of whitefly invasion in cotton. It has become major pest in certain areas under certain conditions. BT cotton still provides complete protection against Heliothrix armigera. However, pests can cause damage to non-BT cotton. With time, certain minor pests have emerged as major ones in BT cotton. I can recall that in year 2005, this whitefly became major pest, particularly in the state of Haryana, Punjab, Rajasthan and Himachal Pradesh. That time it did lot of damage to the cotton crop. So bollworms could not affect it, but whitefly, which was a minor pest, had become major pest that year. With time, certain minor pests have emerged as major ones. Outbreak of invasive mealybug, leafhoppers, thrips, aphid, whiteflies are being experienced. Let's start with jacid, Amresca bigutula. It is called hopper, leaf hopper. Both nymph and adult suck the cell sap from the plant tissues and inject toxin into it, resulting in hopper burn symptom. Means the leaves will dry and it will look that the crop has been burnt. So hopper burn symptoms are caused by jacids. Affected leaves show crinkling and curling symptoms almost all over the plant. Heavy infestation of jacids result in drying of leaves, reduced photosynthetic activity that hampers the productivity of cotton. Means in case of severe invasion or infestation of jacids, the crop may suffer a lot and its productivity can be reduced. Now you can check this picture. You can see on the left side it is adult jacet. On the right side you see infested leaf. There are numerous very small nymph of this jacet. Now after jacet, aphid. Let us see aphid. So far control of all these insects is concerned. We will deal with together. Once we finish the portion of their damage, then we will see how they can be managed. So number 2 major insect is your aphid. Apis gossypi. Damage by feeding on phloem sap. So they also feed on the cell sap. Damage by excretion of honeydew, which attract sooty mold, reducing photosynthesis, and plant vigor and fiber quality by deposition of honeydew on open boughs. So what these aphids do? They release some honeydew kind of excreta, which is deposited on the leaves and other parts of the plant, and it attracts certain fungus. And those fungus can grow on the leaves, and then the photosynthesis area can also be reduced. So they are doing damage, direct damage by sucking the sap, and indirect damage through their honeydew, which is excreta. Leaves crumpling and downward curling. Younger parts suffer more attack than older one. Aggregating populations seen in terminal ends. So large number of adult and nymphs can sit on the terminal part, on the growing part, particularly in case of aphids. Now you can see they are really very small in the picture. You are seeing the bigger one, but it is not that big actually. Picture is taken from a very close point, and you can see on the right side also plants infested with aphids. So here they sit here in large number, particularly in growing parts or terminal parts of the cotton plant. Now the third important group of insect is thrips. It is Thrips palmi or Thrips tabaci. 2 species are there. Nymph and adults suck sap both from upper and lower surfaces of the leaves. So like jacets, like aphids, thrips also suck the sap. They inject saliva and suck lysed content of plant cells, resulting in silvery or brown necrotic spots. So necrotic spots are the dead spots, and they cannot recover back. Once the damage is made, it is made forever. Then reduced growth, loss of vigor, leaves become wrinkled and distorted, curl upward with white shiny patches. So these are typical symptoms of attack by thrips. Rusty appearance in patches develop on underside of leaves. Premature dropping of squares. Squares are the fruiting structure. Delayed crop maturity and reduction in yield. And you can see the picture on the left side of the adult, and on the right side you can see the nymph. So you can see here the difference in nymph and adult thrips. The fourth important pest is your white fly, Bemisia tabaci. As the name suggests, the color of this fly is white. So it is a sucking pest which suck the sap from plant parts, reducing photosynthetic activities and yield. So like your aphid, it also releases certain honeydews which covers certain part of the leaves, and on them some sooty fungus grow, and it is blackened. So photosynthesis is affected. And also transmission of cotton leaf curl disease. This white fly transmits it acts as a vector. So it transmits certain virus diseases like transmission of cotton leaf curl disease. The students can remember that this white fly transmits cotton leaf curl disease from one plant to another plant, acting as a vector. Late season damage severely affects seed development and lint quality. So lint is actually the fiber. The fiber of the cotton is known as lint. Now you can see picture of the white fly. This is adult white fly on the left side. So it is bit bigger in size, but you can see the smaller nymphs. They are very small in size and very large in number, and they can suck the sap from the leaves and other parts of the plant. Now next, number 5 is cotton mealybug, Phenacoccus solenopsis. So in this case also, nymph and adult suck the sap from all plant parts, whether it is leaf, tender stem, twigs or flowers or balls, etc. Stem distortion, twisting and bushiness. Sooty mold develops. Sooty means black. Black mold develops on honeydew. Honeydew is their secretion. Attract ants which act as a carrier of crawlers from one plant to another. Late season infestation cause early senescence. Senescence is kind of maturity, kind of death of the tissues. As a consequence, reduced vigor and significant yield loss. So it can bring your senescence early also. So therefore, overall productivity of the lint may be reduced. You can see this adult female mealybug on the left side, and you can see the accumulation or aggregation of many nymphs on the leaf, twigs on different parts of the cotton plant. They are in large number and they are sucking lot of cell saps. Now, sixth one is papaya mealybug. Of course, this is mealybug of papaya, but it also infests cotton crop. Scientific name is Paracoccus marginatus. Mainly feed on papaya, also infesting cotton crop. Seen scarcely at few locations of central and south India. Damage symptoms similar to cotton mealybug. And you can see on the right side papaya mealybug picture and infested leaf. Next is Indian cotton mirid bug. Scientific name is Creonitates biceratans. Nymph and adults damage developing flower buds and tender boughs. Feeding on flower buds, this is important that they feed on the flower buds, it resulting in oozing out of yellow fluid from the buds and staining of yellow fluid on the inner surface of the bracts. So this is typical symptom of its infestation. Heavy shedding of squares and boughs reduce seed cotton yield. You can see Indian cotton mirid bug picture on the left side and nymph on the right side. Next is number 8, American bollworm, Helicoverpa armigera. Leaves feed tender leaves initially. Larvae feeds tender leaves initially and then bore in a square. Buds, balls and seed with their head thrust into ball. Means they are mostly affecting the reproductive structures of the cotton plant and leaving rest of body outside. Excreta surround entry hole. Damaged squares flare up, have damaged holes on them and are shed. So there may be shedding of the squares. Clear-cut round feeding holes on squares and boughs with or without larva seen. So these are symptoms of American bollworm and it can be sometimes quite damaging and serious pest. American bollworm continues. This pest is currently under control due to wide cultivation of BT cotton but can affect the non-BT cotton. So you can see on the picture it is entering into the reproductive part, flared up damaged square, that is squares. On the right side you can see the larvae. This larvae is entering and making a hole. Through this hole it enters and feeds the internal material of the balls. Next is number 9, spotted and spiny bollworms. Spotted bollworm, it is Aereus vitella and spiny bollworm is Aereus insulana. So these are two species of spotted bollworm of cotton. Terminal shoots dry and withers away when the larvae bore into the pre-squaring plants. Main stem collapses if growing point is affected. Feeding holes in squares and on balls are seen with or without larvae. As you have seen in case of American bollworm, similarly in spotted and spiny bollworm, also there are holes which are seen on the balls through which insect can take entry into it. Flare up of squares and their shedding, premature dropping or opening of attacked balls are common. Means again it can reduce the yield. Larvae do not confine feeding to a single ball, and hence damage is not proportionate to their number. At present, this pest is under control on BT cotton. So that means one caterpillar or one larvae can infest more than one balls. So therefore, the destruction can be very severe. You can see the picture of the destruction of or damage by spotted and spiny bollworms on the right side. You can see even within one ball you can find two larvae. Two larvae can be there. Now, pink bollworm, it is scientifically called as Pectinophora gossypiella. Larvae initially feed on flower buds, resulting in failure of opening and shedding. Roseated flower is typical symptom. Roseated flower infestation during early reproductive growth means the size reduces. Larva enters into developing ball through rind and feed on developing cotton seeds. In younger balls, entire content can be destroyed. In older balls, larvae feed on up to 3 or 4 seeds. Within a ball, more than one larva can be observed. Small exit holes seen on developing balls. Quality of lint deteriorates. Lint is the fiber due to presence of larvae and staining by pest. So it may not remain white. It can change its color, which is not desirable. Pest is seen to damage BT cotton in parts of central and south India. So you can see this larvae feeding inside green ball. So its name is pink bollworm. So you can see the color of the larvae is roughly pink, and you can see on the right side damage green ball by the pink bollworm. 11th number insect pest of cotton is tobacco caterpillar, Spodoptera litura. Its larvae attack in large numbers and they are gregarious in nature. Gregarious in nature means they are found in groups at a certain stage, which is known as gregarious stage. And neonate larvae means newly born larvae after hatching from egg masses feed gregariously by scraping leaves, leaving behind veins and veinlets. So it eats most of the interveinal areas of the leaves. Grown-up larvae feed voraciously and cause heavy defoliation. So sometimes this tobacco caterpillar can be very, very destructive pest in cotton. So you can see gregarious feeding habit of larvae. They are large in number on the left side, you can see in the picture, and size is also very, very small. And they are affecting this leaf, they are eating the leaves. On the right side, you can see solitary larva, grayish in color, and they also damage this crop voraciously. Now coming to the management part of all these insect pests as per growth stage. So you will find that certain insects are confined only to certain stages of the crop, but you will also find that some insects are found throughout the cotton growing stages. So first let us see crop growth stage from 0 to 60. What are the major insect pests in cotton during first 0 to 60 days after sowing? So first one is your pink bollworm. So what can be done to control pink bollworm during 0 to 60 days? Installation of pheromone traps for monitoring population at 45 days after sowing at 5 traps per hectare. So sometimes these pheromone traps are used to detect the injury level of the insect, at which some other measures like use of insecticide can be done. If moth ETL is 8 per trap per night for 3 consecutive nights, mass trapping can be destroyed at the rate of 25 to 30 traps per hectare. Then what can be done is spray neem oil 5 ml per liter plus 5% neem seed kernel extract plus 1 gram detergent powder per liter at 50 to 60 days after sowing. So this is quite environment friendly control or management of this pink bollworm. Then at the 0 to 60 days stage, you will also find some sucking pest. So as the population of natural enemies is abundant in the initial stage that keeps pest population under the check. So, you need not to bother about many of these sucking pests during 0 to 60 days stage, but conserve natural enemy by use of selective insecticides, biopesticides, botanical if required. Spray flunicamid 50 Wg 4 gram 10 liter of water. In case of severity, these kind of treatments can be used. Next is during 0 to 60 days after sowing, tobacco caterpillar, American and spotted bollworm can also affect this crop. So, what can be done? Collect and destroy egg masses, gregarious or solitary larvae by hand picking. So, hand picking can be done. Spray the crop with neem oil at the rate of 5 ml per liter plus 5% neem seed kernel extract plus 1 gram detergent powder per liter at 50 to 60 days after sowing. Under emergency situations, spray chlorantraniliprole 18.5 SC at the rate of 3 ml per 10 liter. So, insecticide is also available that can also be employed. Now, let us see what are the insects during 60 to 90 days after sowing. So, again this pink bollworm can come even during this stage. So, monitoring population buildup by observing rosette flowers or dissecting green balls under field conditions. Such flowers need to be plucked and destroyed. Means you can check the flowers, you can check the initial balls. If you find some larvae there, then those kind of balls or flowers should be destroyed or removed from the field. If ETL, economic threshold level, is crossed 10% infested flowers or balls, spray Cunalfas. Cunalfas is an insecticide, 20% AF at the rate of 20 ml per 10 liter of water. Now, during 60 to 90 days, jassids and fall armyworm can also affect this crop. For jacid, if more than 25% plants showing symptoms, spray flunicamid. Flunicamid can be sprayed. This is 50 Wg at the rate of 4 gram per 10 liter of water. You can remember this name, flunicamid 50 Wg. Then fall armyworm, spray crop with neem oil 5 ml per liter plus 5% NSKE, neem seed kernel extract plus 1 gram detergent powder plus 1 per liter of water, or spinotram 1.7 SC at the rate of 8 ml per 10 liter of water. So, you can see whenever we use these neem based pesticides and many other pesticides, we use this spreading agent, this detergent powder. It acts as a spreading agent so that your spraying material can spray longer on the leaves or it can stick to the leaves. Therefore, its effectiveness is improved. Next is during 60 to 90 days, next is whitefly. So, what can be done to control? Install yellow sticky traps for monitoring and management of whitefly. As you are aware that it will suck the sap of the leaves and other parts. So, if whitefly crosses ETL, economic threshold level, spray buprofezin 25% SC at the rate of 10 ml per 10 liter. This SC is soluble concentrate. This is a kind of formulation for certain pesticide. Mealybug, for mealybug, destroy Congress grass, remove infested plants and destroy and conserve the parasitoids. So, this is how we can manage mealybug. Another insect pest can come, that is bollworm or American and spotted. Both kind of bollworms can come to the cotton crop at 60 to 90 days after sowing stage. Damaged square are more than 20% with bollworm infestation. Then we can spray the insecticide, which is chlorantraniliprole, very long name, chlorantraniliprole 18.5 SC at the rate of 3 ml per 10 liter of water. Then during 90 to 120 days after sowing, pink bollworm, you see it is coming again. It is coming again and again. So here what can be done? Release parasitoid trichogramma bacteria at the rate of 1,50,000 per hectare. So this is biological control and quite effective also. Trichogramma bacteria. If population crosses ETL, spray profenophos 50% EC. This EC is emulsifiable concentrate kind of formulation of insecticide at the rate 30 ml or imamectin benzoate 5% Sg 5 gram per 10 liter water after 10 days of parasitoid release. So this is important. You should not spray any of these insecticide immediately after application of the parasitoid. At least 10 days period should be there. Now, American and spotted bollworm can also come at 90 to 120 days stage. Here you can see if more than 20% plants have been flared up, squares, spray indoxacarb 14.5 SC at the rate of 5 ml or spinosad 45% soluble concentrate at the rate of 2.5 ml per 10 liter of water, and needful solution of the insecticide can be prepared and sprayed on the crop. Jacids and thrips. If more than 25% plants show symptoms of jacids on underside of leaves by thrips, spray thiamethoxam 25% Wg at the rate of 2 gram per 10 liter of water. That means you need to take 2 gram of this insecticide and dissolve in 10 liter water, and in this proportion or in this ratio the desired solution can be made and then sprayed on the crop. Now, whitefly during 90 to 120 days, if population crosses ETL, economic threshold level, which is 6 whitefly per leaf. At this particular stage, the ETL of whitefly is 6 whitefly per leaf. Spray dinetofuran 20 Sg at the rate of 3 ml per 10 liter of water. Then fall armyworm, spray spinetoram 1.7 SC at the rate of 8 ml per 10 liter or chlorantraniliprole 18.5 SC at the rate of 3 ml per 10 liter of water. Now see pinkworm, if growth stage is more than 120 days after sowing, then pinkworm can again come. If more than 10% of balls are infested with pink bollworm, then you can spray insecticide like fenvalerate 20% emulsifiable concentrate at the rate of 10 ml, or you can use cypermethrin 10% EC at the rate of 10 ml per 10 liter, or you can say 1 ml per liter of cypermethrin can be used. Now you have seen that number of insect pests are affecting our crop, cotton crop, and you have also seen their control measure. The emphasis most of the time for control of insect pests is given on crop rotations, on cultural management, on clean cultivation, on biological methods and so on. And the use of insecticide should be the large resort, and in many cases like neem based pesticides are also available that can be used, because these chemical pesticides may be harmful to our soil and can be harmful to our environment if their doses or frequency increases. So therefore, we should try to follow integrated pest management strategies in cotton crop. Let us check main diseases of cotton crop and how these diseases can be managed. Now first disease is root rot. So it is caused by fungus Rhizoctonia solani. Many of you might be knowing that Rhizoctonia solani mostly lives in soil. So kind of soil borne fungi, or bataticola means Rhizoctonia bataticola, and another species is Sclerotium rolfsi. So these are the important species of fungi that can cause root rot in cotton. Drooping of leaves and wilting. Drooping means these leaves actually hang. They are just hanging on their branches. Infected plants can be easily uprooted. Lack secondary roots and discoloration of roots is seen. Next is fusarium wilt. Like insect pests and diseases, we will also see control measures of all the diseases together. So now you check the symptoms of fusarium wilt. So caused by fungus Fusarium oxysporum, and subspecies is Vas infectum that can appear at any stage of plant development. It can come at early stage, middle stage, or even at later stage of the cotton growth. And what happens? Loss of turgidity, yellowing and browning of foliage followed by death of the plants, and dry soils favor infection. This is important. Then ball rot, third important disease. It can be caused by Coletotrichum gossypii or gossypii. So you can remember scientific name of this fungus, Coletotrichum gossypii. Other is Diplodia gossypina, Ascochyta gossypii, and Fusarium species. So number of fungal species can cause this ball rot. Deformity and immature ball opening. Drying and visible fungal growth on infected balls. So ball can rot down actually. And ball rots first appear as water-soaked spots in case of bacterial infections. In fungal infection, they are generally covered with a fungal growth. So quite easily distinguishable. So fungal growth can be seen on the balls, and one can make distinction in fungal and bacterial infection. Now you can see the rotting of the balls on the left side. So you can clearly see the fungus mycelia on infected balls. It can be seen through the naked eyes also, you can check. And then internal ball rot. You can see the rotting of the ball is happening. So it is affecting the quantity as well as quality of the lint. Now next disease is Alternaria leaf spot. As the name suggests, Alternaria is fungus and leaf spot means initially it gives some spots. So it is caused by Alternaria gossypina, Alternaria macrospora, and Alternaria alternate. So it kills surrounding tissues and produces more spores on lesions quickly. Spores more severe on lower leaves than upper leaves. So this is important, one can check. So on the right side you can see the typical lesion or spot of disease. So such kind of spots, browny spots are there on the leaves. You can see more clearly on the second leaf also. They are large in number, these affected leaf by Alternaria leaf spot. Now let us check grey mildew caused by Ramularia areola. Irregular, angular, pale, translucent spots measuring 1 to 10 millimeter in size surrounded by veinlets. You can see on the right side the whitish, the mildew kind of growth on the leaves. Appears on older leaves, usually when plants reaching maturity. A frosty or mildew growth consisting of conidiophores of fungus appear on leaves. So on the right side you can see the variety of symptoms produced by grey mildew. Now bacterial leaf blight caused by Xanthomonas axonopodis PV malvacearum, also known as angular leaf spot. Angular leaf spot or bacterial leaf spot. What happens? Seedling blight, blackening of stems, leaf spots and ball rot. So symptoms can come at different parts or different stages also. A small angular water-soaked spots on underside of young leaves, and then spots turn brown to black as infection progresses. So you can see the spots on the right side on these leaves. Now cotton leaf curl disease CLCUD transmitted by white fly, small vein thickening symptoms on young leaves, Upward or downward leaf curling means on the right side picture you can see the curling of the leaves. So, curling of the leaves can be upward and can be downward also. Upward or downward. Severely affected young plants are stunted and often do not bear any fruiting bodies. So, this can be quite damaging many times. Now, management of cotton diseases, we will deal in totality how to manage these diseases. So, preventive measures are the best. So, they are followed before the sowing and at sowing. So, field should be deeply plowed and left for solarization to kill soil-borne pathogen. So, this is a practice also, deep summer plowing. It kills variety of inoculum, variety of insect pests, their eggs and so on. So, it is quite useful, this summer plowing. Fields with long history of fusarium wilt or root rot should be avoided for growing cotton. Many times what happens, farmers follow monoculture of cotton. Year after year, they take cotton crop in the same field. So, therefore, definitely you will have more diseases under such condition. So, better to rotate the crop. For at least 2 or 3 years rotation should be followed. 3-year rotation means for 3 years. In 3 years you can take cotton crop only once, and then after 3 years you will take cotton in the same field. So, at least 2 to 3 year rotation is good to avoid many diseases. Do not allow irrigation water to flow from root rot affected fields to healthy fields. So, some diseases can be transmitted through the water also. So, therefore, water movement can be restricted. Apply field to field. Avoid monocropping and cultivation of cucurbitaceous and solanaceous crop in adjoining fields. So, they can attract some insect pests. So, better to avoid cultivation of cucurbitaceous and solanaceous crops around the cotton fields and remove the weeds in and around fields, because these weeds can act as host to some insect pests and diseases or alternative host. Now, you see chemical and biological control of diseases after preventive measures. Now, root rot. So, what can be done? Apply zinc sulphate at the rate of 24 kg per hectare. It helps, it gives better tolerance to the disease. Seed treatment with trichoderma. Trichoderma is biological agent which kills fungi or reduces fungal growth. So, trichoderma at the rate of 4 gram per kg seed, or Pseudomonas fluorescens. So, these are bacteria which are also they stop certain diseases at the rate of 10 gram per kg seed, or soil drenching can also be done with trichoderma at the rate of 10 kg per hectare, mixing with 200 kilogram of moist farmyard manure. So, drenching means kind of wetting in a specific pocket of the soil, so that these organisms reach in the nearby area of the roots, and they can control certain diseases in and around the roots. Fusarium wilt. For this, seed treatment with therum 75% WS at the rate of 3 gram per kg seed, or spot drenching with carbendazim 50% WP at the rate of 2 gram per liter water. So, spot drenching of carbendazim or bavastine can be done at the rate of 2 gram per liter of water. So, in this case you are just putting solution of the fungicide near the roots of the plant, or wetting the soil through the drenching process. Bole rot. Spray carbendazim 50% WP at the rate of 2 gram per liter water. For internal bole rot, spray with streptocycline 2.5 gram plus copper oxychloride 25 gram per 10 liter of water. Means you need to use a mixture of bactericide as well as fungicide to control bole rot. So, in this case what we can do, we can take 2.5 gram of streptocycline and 25 gram of copper oxychloride in 10 liter of water. The desired solution can be prepared and sprayed on the crop. Alternaria leaf spot. Spray pyraclostrobin 20% Wg at the rate of 2 gram, or Metiram 55% plus pyraclostrobin 5% Wg 20 gram per 10 liter of water. So, two options are available. One can straightway go for single chemical, that is pyraclostrobin, or you can use combination of two chemicals. One is Metiram and other is pyraclostrobin. For bacterial leaf blight, seed treatment with carboxin 75% WP at the rate of 1.5 gram per kg seed, and foliar spray with streptocycline 1 gram plus copper oxychloride 25 gram per 10 liter of water. So, that can control or reduce the ill effect of bacterial leaf blight in cotton. Now, you have seen how these insect pests and diseases are controlled. Just now you have seen that number of measures are available. Among all these measures, the preventive measure should always be preferred for the management of diseases also. And integrated approach or integrated disease management is the best way, where we can combine some cultural approaches, mechanical approaches, or some biological approaches or biological methods, biocontrol methods, or crop rotations, and at the end we can use some chemicals also if it is desirable. Now, after seeing these plant protection measures, now almost your crop is matured now, and now you need to go for harvesting of the crop. So, there are number of methods for harvesting of the crop or cotton crop. Manually it can be harvested by labor, using labor, or it can be harvested by machine. But when you are going to harvest this crop by machine, then you need to defoliate the crop. You need to remove the leaves of the crop. So, we will discuss in detail what are different harvesting method of cotton crop. So, the entire cotton in India is hand picked. In our country, the introduction of machine is minimum, very minimum. About 450 to 1500 man hours per hectare with an average of 0.9 man hours per kg of cotton. So, you can see in the harvesting or picking, the harvesting of cotton is actually picking if you are doing it manually. Large number of laborers are involved, which can range from 450 to 1500 man hours per hectare. In recent years, labor shortages appear during the peak periods of cotton harvesting. So, pickings delayed, causing field losses. So, you cannot finish picking job or harvesting job so quickly. So, what can be done? We need some alternative, which is machine. Also, hand picked cotton is considered to be cleaner than those picked by the machine. Definitely there are limitations in manual harvesting as well as in mechanical harvesting. Harvesting by machine may lead to reduction in the quality. However, the hand picked cotton has better quality. So, number of machines are available for commercial harvesting of cotton crop in the market. So, there may be two kind of machines. One is spindle type and other is stripper kind of harvesters. So, the entire cotton in the USA, Brazil, Australia, Israel, Turkey, Argentina is picked by machines. So, in most important countries it is picked by machines, but in developing countries like India it is mostly hand picked. The spindle type pickers are selective in that the seed cotton is removed from the open balls, whereas green or unopened balls are excluded. So, in this case you can see that the seed cotton is removed from the open balls. So, what is seed cotton? Seed cotton is actually fiber plus seed, fiber plus seed. These machines have spindles that twist and pick the seed cotton from the burr, that is capsules, attached to the sympods. Spindle type pickers are usually designed for a row spacing of 97 to 102 centimeters. Strippers are another group of machines, on the other hand, are once over. All the balls are picked in one operation. Machines and can operate at narrow row spacing. So, these are the differences, that it can collect all the boughs. Cotton harvested by brush strippers generally had high moisture content than spindle type pickers. Although the picking efficiency is high, machine picked cotton has a major drawback of high trash content. So there may be more trash or residues content. Furthermore, green leaves have to be removed to facilitate mechanical harvesting. So this is extra operation that you need to do to accomplish mechanical harvesting. So defoliator actually facilitate uniform ball maturation and induce leaf drop. Cotton defoliation is complicated process and must be carefully timed and executed. Two types of defoliant or their combinations are used. The defoliants may have herbicides with contact activity or hormones with growth regulatory effect. So two options are available for defoliation, means removing the leaves. Tribuphos and endothal are from the first category, means kind of herbicides. They cause plant injury, and the injured plants in turn produce more ethylene, which induces the abscission. Abscission means removal of leaf from the twig or from the branch and dropping of the leaves. Hormonal defoliants like thidiazurone or ethepone induce in situ ethylene production. As many of students might be knowing that it is maturity hormones. It brings maturity early, ethylene, leading to the dropping of leaves and shedding of balls. Ball opening chemicals are often used in combination with defoliants to accelerate ball opening and increase the percentage of open balls for mechanical picking. Now, let us see post-harvest management and quality of the cotton fiber. So, commercial portion of the produce, which is lint, is fiber, has to be separated from the seed. So, lint has to be separated from the seed. The process of separation of seed and lint is called as ginning. Traditionally, manual methods of ginning were employed that were slow and tedious, means people were removing seed and fiber by hands. Cotton gin, invented by Eli Whitney in 1793, paved way for commercial importance of the crop, means then machine was invented. Ginning percentage, it is also known as ginning outturn and expressed as the ratio of lint is fiber to seed cotton, means seed plus fiber, expressed in percentage. Range from 28 to 30% for Barbadans cotton, 33 to 38 for hirsutums, and 36 to 42% for Desi cotton. So, you can say generally Desi cotton has high ginning percentage. After ginning, the lint is pressed into bales, and the standard weight of a bale in India is 170 kg. You can remember 170 kg is the weight of one bale in India. Now, fiber quality. Fiber quality is a varietal character, though environmental factor like climate, soil moisture, available during growth and proper use of fertilizer also affect the fiber quality. So, major characters that determine the commercial value of cotton fiber are color, length, fineness, strength and spinning performance of the fiber are the important characters. Like color, cultivated cottons are white in color. People prefer white color. However, colored cottons are also available like green, brown, blue of various shades. Then come to the length. Length is another parameter. It is the most important among fiber properties from an economic point of view. Halo length and staple length is the arithmetic mean of length of all fibers present in a sample of cotton. Then you can see the length and classification of the cottons on the basis of staple length. Short 20 millimeter or less, medium 20.5 to 24.5, and then medium you can see 25 to 27 millimeter, long 27.5 to 32, and extra long more than 32.5 millimeter. Then comes the fineness. Fineness is cotton quality parameter. Next is importance only in the fiber length. It is like a genetic trait, and you can see the classification. less than 3 millimeter very fine, 3 to 3.9 fine, average 4 to 4.9, coarse 5 to 5.9 and very coarse more than 6 millimeter. Strength. The durability or wear life of textile material depends greatly on the mechanical strength. Fiber strength is expressed in terms of tenacity, defined as the load or force required to break a fiber of unit linear density, gram per tex or centinewton per tex. Then spinning performance is measured by number of counts. Counts refer to the number of hanks, each of 840 yards, which weighs 1 pound. Today we have varieties with a count range from 6s to 126s. The oil content in cotton seed is crushed in order to separate its 3 products, oil, meal and hulls. So, oil content may vary from 14 to 14.6 to 20.6%. Cotton seed oil is now the 3rd major oil that is consumed in India. But you need to remove some toxic material to make it usable by human beings. It is also used, this cotton cake is quite useful for the animals and high protein concentration can also be made. Coming to cropping system, this cotton is grown as a sole crop, as an intercrop also. So, in northern state where double cropping is followed, cotton is planted after the harvest of wheat or mustard. In dry regions, central India, cotton was traditionally rotated with sorghum. Presently, monocropping is a common practice after the BT cotton was introduced. Rotation with soybean has replaced sorghum due to its better market price. Irrespective of input management, continuous cotton cultivation leads to decline in productivity. Strip intercropping of cotton and pigeon pea is in replacement series is most prevalent in central zone or central parts of India. It also intercropped with soybean. Number of legume or oilseed can be intercropped in the cotton crop. Crop like chili, onion can also be intercropped. So, you can see different crop sequences and intercropping systems in different states. For example, in Madhya Pradesh you can see cotton, cotton jowar, cotton wheat, and intercropping you can see cotton black gram and cotton plus soybean. Then in Maharashtra cotton monocrop, cotton jowar. In intercropping you can see one can take green gram in Maharashtra, soybean and groundnut. And then in Karnataka you can take chili, onion or rice as an intercrop. In Tamil Nadu, onion, groundnut and black gram. And cropping system in Tamil Nadu can be rice cotton, rice cotton, cotton jowar, cotton pulse jowar. In Andhra Pradesh you can see cotton rice, cotton chili and cotton tobacco are the rotations, and for intercropping cotton plus black gram, cotton plus pigeon pea, cotton plus chili, etc. Dear students, I hope that you have enjoyed this lecture. Thank you very much.